Welcome. In this section, we will dive into Listeria species, exploring its pathogenesis, virulence factor, the key risk factors that make it such a significant pathogen, and the laboratory diagnosis. Let's get started. Listeria belong to the family Listeriaceae, including several species such as Listeria monocytogenes, which is a type species, and Listeria ivanovi. Morphologically, Listeria are gram-positive short rods, and they are non-spore-forming and non-acid fast. These organisms are facultative anaerobe, and the growth is enhanced in the presence of 10% carbon dioxide. Coming to their biochemical characteristic, Listeria are catalase positive and oxidase negative, and they are capable of hydrolyzing esculin. In culture, they grow well on blood agar, where pathogenic species like Listeria monocytogenes shows beta hemolysis. However, they do not grow on McConaughey agar. A key identifying feature is their motility. Listeria exhibit a trembling motility at 28 degrees Celsius due to the presence of their peritrichous flagella. Another important feature is their ability to grow in a wide range from 4 degrees Celsius to 37 degrees Celsius, making them psychotrophic and capable of surviving refrigeration. Additionally, they can tolerate high salt concentration up to 10% sodium chloride. These features make Listeria highly adaptable and significant in both veterinary and public health contexts. Among the Listeria species, Listeria monocytogenes is the most important pathogen. It causes a range of serious conditions including septicemia, abortion, mastitis, and central nervous system infection such as encephalitis, especially in ruminants. Listeria ivanovi is also pathogenic, but mainly affect animals. The other species are generally considered non-pathogenic. Talking about habitat, Listeria is widely distributed in the nature. Its primary habitat is soil and decaying vegetation, where it survives as a saprophyte. Silage is the main source of infection in the case of ruminants, particularly when the pH is above 5.5, allowing the bacteria to multiply. It can also be found in water, sewage, animal feed, and a variety of food products, including raw milk, cheese, meat, and seafood. From a public health perspective, Listeria monocytogenes is a significant foodborne pathogen. Common sources of human infections include soft cheese, deli meats, milk, dairy products, as well as undercooked poultry. An important point to remember is that both humans and animals can act as an asymptomatic carrier, contributing to the spread of infections. Listeria monocytogenes affect a wide range of hosts and produce multiple disease conditions. In animals, it commonly causes septicemia, also known as the visceral form, often associated with the necrotic foci in the liver. You can see white spots on the liver. It's a necrotic foci, and also in other organs also. In ruminants, such as sheep, goat, and cattle, it causes the neural form or the circling disease. It is characterized by meningoencephalitis with a micro abscess and perivascular cuffing in the brainstem region. In pregnant animals, such as especially in the case of sheep, goat, and cattle, Listeria lead to abortions, typically during the 3rd trimester. In cattle, it can also cause mastitis and iritis, particularly associated with the silage feeding. In humans, Listeria monocytogenes is an important zoonotic pathogen causing meningitis, septicemia, gastroenteritis, and cutaneous lesion. Listeria ivanovi, in contrast, primarily affects sheep and cattle, which is mainly associated with abortion. These disease patterns are important for both diagnosis and epidemiological understanding. Listeria infections are influenced by several key risk factors. Silage feeding, especially when the silage pH is above 5.5, create an environment where Listeria can multiply and survive. The winter season increases risk as animals are often fed most of feed and are confined indoors. Finally, high iron content in the feed and water can promote bacterial growth, enhancing susceptibility to infection. Understanding these factors help in preventing and managing Listeria outbreak in livestock. Listeria monocytogenes uses several virulence factors to infect the host cells and spread. The first one is internalins, including internalins A, B, and C, help the bacteria to attach to and invade the host cells. Next is Listeriolysin O or LLO. It's allow Listeria to escape from the phagosome into the host cell cytoplasm. Next is phospholipase C, which help aid in phagosome escape and help the bacteria spread by lysing the cell membrane. Phosphatidylinositol specific phospholipase C is a key Listeria virulence factors with the lecithinase activity. It helps the bacteria lyse the phagosome membrane, allow escape into the host cell cytoplasm. Additionally, this factor mediates bacterial cell-to-cell spread and promoting the infection of the neighboring cells, sustaining Listeria's intracellular life cycle. Next is ActA. It drives actin-based intracellular movement, letting Listeria propel itself within and between cells. ActA. Next is PRFA. It is a master regulator controlling the expression of many virulence genes. Metalloproteins activate the phospholipases for the effective host cell invasion. Next is IAP or P60. It contributes to the cell invasion, while fluctuating or DECU, it will regulate the motility and the overall virulence. Together, these factors make Listeria a highly efficient intracellular pathogen. Next, we look into the diagnosis of Listeria. Laboratory diagnosis of the Listeria depend on the disease form. For septicemic form, we have to collect the samples like liver, spleen, and kidney are sampled. Neural infection require the brainstem, medulla, and the spinal fluid. In abortion cases, placenta, amniotic fluid, and fetal tissues are submitted. Feed sample, especially silage, can also help track infection sources. The specimen should be cultured promptly at 35 degrees Celsius or stored briefly at 4 degrees Celsius. For long-term preservation, freezing at -20 degrees Celsius prevent overgrowth of contaminant. Always collect sample aseptically. Direct smears may reveal gram-positive rods, but isolation is necessary. Histopathological examination of the formalin-fixed brain tissues can provide a presumptive diagnosis for neural listeriosis due to its distinctive lesion like microabscess or perivascular cuffing. Isolation of Listeria remains the gold standard, combining selective enrichment with colony characterization. Clinical specimens are usually inoculated on the blood agar with Makongi agar used for contaminants. Neural cases may require cold enrichment at 4 degrees Celsius for up to 12 weeks, favoring Listeria growth over other bacteria. The selective enrichment broth include LEB, Fraser, UVM, and talcum help isolate Listeria from complex samples, and also other selective agars like OX, MOX, LPM, and Chrome agar are used. Listeria are used to grow and differentiate colonies. For faster detection, antibody or molecular based test we have to conduct. If we look into the blood agar, this Listeria ivanovi will produce a wide zone of beta hemolysis resembling the beta hemolytic Streptococcus species. In contrast, Listeria monocytogenes and other non-pathogenic Listeria produce a narrow zone of beta hemolysis on blood agar. So this we can use for differentiation of Ivanovi and monocytogenes on blood agar. And another feature is that this Listeria species, when grown in a semi-solid motility media, it shows an unusual umbrella-shaped growth in the subsurface. Listeria colonies are small and smooth on blood agar. Microscopically, they are short gram-positive rods and non-sporing also. The key identification test is catalase test, that is catalase positive. It will hydrolyze esculin and characteristic tumbling motility at 25 degrees Celsius and umbrella-shaped growth on semi-solid media. And we can also conduct CAM test, modified CAM test, will help to differentiate Listeria species. Listeria monocytogenes shows enhanced hemolysis with the Staphylococcus aureus. Here you can see Listeria monocytogenes enhanced beta hemolysis with the Staph aureus. But Listeria ivanovi is enhanced with the Rhodococcus equi, not with the Staph aureus. Careful interpretation is needed as some interactions may overlap. So for Listeria species differentiation, like Listeria monocytogenes and Ivanovi or Ivano subspecies londiniensis, Listeria monocytogenes have a narrow zone of beta hemolysis, but the Ivanovi have a broader zone of hemolysis. In the CAM test, monocytogenes give positive with the Staph aureus, but Ivanovi will give positive with the Rhodococcus equi. So sugar fermentation, Ivanovi give positive with the ribose and with the D-xylose. Monocytogenes is positive only for L-rhamnose, but Listeria ivanovi subspecies londiniensis give positive for D-xylose. Pathogenicity testing for Listeria. The pathogenicity testing evaluate the virulence of Listeria species using laboratory models. The Anton test involves inoculating the conjunctiva of rabbit and guinea pig, where only Listeria monocytogenes cause purulent keratoconjunctivitis within 24 to 36 hours. Next is in mouse intraperitoneal inoculation. Both Listeria monocytogenes and Ivanovi are pathogenic, causing death within 5 days with a necrotic liver lesion. Other models include inoculation of CAM in embryonated eggs, sensitive immunocompromised mouse models, and CACO2 human intestinal epithelial cell cytotoxicity assay is used to study virulence in vitro.